Now, let me introduce the part 4, classification of molecular imaging. Classification of molecular imaging include radionuclide imaging, sometimes we call nuclear medicine imaging. They will use positron emission tomography, we call PET imaging. Single photon emission computed tomography, we call SPECT imaging. And second is magnetic resonance imaging, we call MR imaging. And the third, ultrasound imaging, we call ultrasound US imaging. And finally is optic imaging, sometimes we call OI imaging. The first, radionuclide imaging. Nuclear medicine imaging is a process of reflecting a specific molecular event in vivo by means of highly specific combination of radionuclide labeled molecular probes and target molecules through nuclear detectors. The main device is PET, and the second is SPECT SPECT. It is a mature molecular imaging technology at the present. It is a classical functional and metabolic imaging with high sensitivity, quantitative, and animal experiment result directly extended to clinic advantage. It used in the three areas. In clinical research, nuclear molecular probes are mainly used to diagnose and study disease such as tumor and the nervous system at the molecular level. In basic research, such as evaluating a transcription and translation of endogenous or integrative genes, evaluating the synthesis, metabolism, and transport of substrate, and evaluating the response of ligand receptors between cells. And in drug research, it provides a new technology platform for the research of a new drug. It can directly observe the drug molecular dynamically, continuously, and repeatedly in vivo experimental animal or animal model. In nuclear medicine imaging, PET, positron emission tomography, or PET-CT, PET-MR, is the most advancing functional imaging equipment now. It can evaluate metabolic and biochemical functional information such as blood flow, metabolism, protein synthesis, gene expression, tissue oxidation, and so on by non-invasive means. The most common used radioisotope is fluoride-18, and this radionuclide is commonly used, account for 90% radiopharmaceutics in clinic. Its half-life only 110 minutes. Now, not only PET or PET-CT, PET-MR for clinic, and also for animal experiment, equipments already existed. We called animal machine, animal PET, animal or micro PET, micro PET-CT. For SPECT detection, they use a single photon radionuclide, such as Technetium-99M, and the half-life only 6 hours and pure gamma ray. And for SPECT, they can be divided into 1 camera, 2 camera, and 3 camera. The more camera, the better. Also, the equipment for animal application has been used for scientific research. Also, we call it microSPECT or animal SPECT. The two modality for molecular imaging is MR, we call it magnetic resonance imaging. They use a magnetic resonance imaging technology and biochemical characteristic of contrast agents, combining to directly or indirectly reflect the normal or the pathological molecular process in living cells. Molecular-level MR imaging uses special molecules that can be visualized on MR imaging as imaging markers to locate the molecule in vivo. Targeted contrast agents based on the gadolinium or GD or superparamagnetic iron oxide SPIOS can be used to measure molecular markers of unknown tissue or their related biochemical information. MR, the relatively high capacity, and the for non-interriding binding site or interriding site, they both can be used. The third, ultrasound imaging. Ultrasound imaging creates 3D image using reflected or transmitted ultrasound, especially at interface, currently used mostly for anatomic imaging, blood flow, tissue characterization or contrast possible, and increasing sophisticated. Target ultrasound contrast agent can be constructed by the linking target molecule, or for example, specific antibody or ligand, to the interface of acoustic contrast agents, which can actively bind to the target area for specific ultrasound molecular imaging. Microbubble contrast agent was introduced into target tissue through blood vessel, and the imaging of target area at tissue level, cell level, or subcellular level was observed to reflect changes of molecular basis on the lesion tissue. Ultrasound microbubble can be used to detect change in cell and molecular level in the early stage of disease, which is conducive to the earlier and more accurate diagnosis of disease. These pictures showed a design of microbubble for targeted drug delivery. In the core, filled with gas, outer, surrounding with an outer shell. On the surface of a shell, linked with a ligand. So when this bubble delivers into body, they will migrate to the targeting area to bind a specific receptor. Then, under the effect of ultrasound, they will deliver the drug into the cell. The fourth, optic imaging. Optic imaging in vivo mainly uses bioluminescence or fluorescence technology to label cell or gene. Using a very sensitive optic detection instrument, we can directly and real-time observe the cell activity or gene behavior of the labeled cell and gene in vivo. Different components are identified by different absorption and scattering of light by soft tissue, and the function information can be obtained by the unique absorption and nature chromophores. The device can observe the growth and metastasis of tumor in vivo and the expression of specific gene in vivo. Fusion imaging with a corresponding imaging process system, the atomic function and the metabolic image of the detected object could be fused, which divided more practical new field of clinical medicine. Fused imaging actually by the different machine detection. Finally, we will talk about a multimodality imaging. Different imaging detector put together on the same machine. They have, for example, PET-CT. PET and CT put together, we call it PET-CT. PET-CT is used to display the physical metabolism function, and CT to show the structure of the human tissue. So CT shows structure, and PET shows the physiological metabolic function change. It is the most ideal combination of examination instrument in molecular imaging. They accurately located the distribution of this nuclide by using cold machine CT technology to provide anatomical and functional information at the same time. Also, PET-MR can collect information of PET and MR simultaneously and acquire the omnidirectional information about the human anatomy, function, and metabolism. The image quality and soft tissue resolution of whole PET-MR are higher than those of PET-CT. It is of great value for early and accurate detection of lesion, accurate localization, and guide clinical treatment. And also, we have the PET SPECT optic in the same machine. And optical imaging has the advantage of no radiation, good stability, and suitable for repeated imaging as a single injection in vivo, especially with the development of optical signal detectors and successfully development of new fluorescence material with stronger tissue penetration, such as biofluorescence. It has become an important branch of molecular imaging. And finally, let's compare the different imaging modality. See this picture. You can see the advantage of radionuclide target imaging, or we call nuclear medicine imaging. They have the advantage in the low density site, lower than 20 nanomole. Okay, that's all for this part. Thank you.